Macromolecules. Most macromolecules are polymers. There are four classes of large biological molecules, which are carbohydrates, lipids, proteins, and nucleic acids. What are polymers? Polymers are long molecules consisting of many similar building blocks, and these building blocks we call it as monomers, which is the small building blocks of polymers. These monomers will be connected to one another through the formation of covalent bonds between the monomers to form polymers. There are three of the four classes of macromolecules which are polymers. They are proteins, carbohydrates, and nucleic acids. All these macromolecules, all these three macromolecules are composed of monomers. The monomers for proteins are amino acids. There are 20 different types of amino acids. Carbohydrates, the monomers are monosaccharides, and the nucleic acids, the monomers are nucleotides. The first type of monomers are the amino acids. There are 20 different types of amino acids. These amino acids are connected together through the formation of covalent bonds between the amino acid, we call it as peptide bonds, to form polypeptide chains. These polypeptide chains will have specific function. For example, they can function as enzyme, which function to catalyze chemical reaction, or the protein can function as defensive proteins such as antibody or immunoglobulin to protect the infections of diseases or virus or pathogen that enters into the body. And then the third function of protein is that it can function as storage protein to store amino acid, which is storage of amino acids such as albumin of eggs. And then the protein can function as transport protein, which is to transport substances across plasma membrane, for example. And then protein can also function as hormones which coordinate organismal responses, such as, for example, hormones that are made up of protein is insulin and glucagon that controls blood glucose levels. And then the protein can also function as receptor proteins which receive signals from outside cells. Later in chapter 3, we are going to look at the different functions of protein that are embedded in plasma membrane. One of it is to function as a receptor for hormones. Okay, so these receptors are bound to the plasma membrane or are embedded in the plasma membrane of cell. And then protein also can function as motor protein, which function in cell movement. So the detail of the function of motor protein we are going to look at in the second chapter, chapter 2, which is cell. And then the next one is structural protein to provide structural support, such as, for example, collagen or keratin that are found in your hair or in your skin or nails. The second types of monomers are the monosaccharides. Okay, so monosaccharides are monomers of carbohydrates. Carbohydrates are classified into three groups, which is the monosaccharides. Examples are glucose, fructose, galactose. The second groups of carbohydrates are the disaccharides, which are lactose, sucrose, and maltose. And the third group of carbohydrates are the polysaccharides, which are cellulose, starch, glycogen, and also chitin. Cellulose, cellulose is the component of plant cell wall. Starch is the carbohydrate storage in plant. Glycogen is the carbohydrate storage in animals, which is stored in the liver. And chitin is the component of exoskeleton of insects and also fungal cell wall. And as for the monosaccharide and disaccharides, they can function as fuel, okay, fuel that will be used during cellular respiration to generate energy for cell in the form of ATP to do cellular activities. Okay, so the next class of macromolecules are lipids. So remember, lipid is the one molecule that is not a polymer. Okay, lipids are not polymer and they do not have any monomers. Okay, monomers. So there are three for your syllabus. There are three types of lipids that we are going to look at. The first one are the triglycerides or triacylglycerol, which exist in two forms, whether in the form of saturated fats or unsaturated fats. Unsaturated fats are usually oils or vegetable oils that exist in liquid form. Okay, so animal fats, they are usually solid at room temperature. Triacylglycerol is made up of two components, which is glycerol and also three fatty acids. The bonds between glycerol and fatty acid, we call it as ester bond. That is the first type of lipid. The second type of lipid or group of lipid is phospholipid. There are two portions of phospholipid, which is the hydrophilic head. The hydrophilic head contains phosphate and a choline group, and it is hydrophilic. Okay, the head is polar and it is attracted to water molecule, and the other region of a phospholipid molecule is the hydrophobic tail, which is made up of two fatty acids. Okay, so basically phospholipid is made up of glycerol, which is bonded to a phosphate group, and two fatty acids. So this phospholipid molecule is hydrophobic, and uh, when you place or throw phospholipid in an aqueous solution, the phospholipid molecules will be attracted to one another, whereby the hydrophobic tails are attracted to one another to form hydrophobic interactions, and the hydrophilic heads region of the phospholipid molecule is attracted to the aqueous surrounding. The third, the third type of lipid is steroids. Okay, so steroid, as you can see, it is composed of four fused ring attached to chemical groups. Okay, so the most common steroids are cholesterol. Okay, so cholesterol is one of the components that are found in cell membrane of animals. And then the cholesterol is also function as precursor molecules that is used to synthesize steroids hormones, such as, for example, sex hormone, such as estrogen or progesterone or testosterone. So all these hormones, initially they are synthesized from cholesterol, which is a type of steroids. Okay, so the last type of monomers are the monomers for nucleic acids, which are the nucleotides. The component of a nucleotide consists of a pentose sugar, okay, 5-carbon sugar, bonded to a nitrogenous base, and the sugar is also bonded to a phosphate group. The nitrogenous base can be adenine, guanine, cytosine, thymine, and uracil, depending on the type of nucleic acids. There are two types of nucleic acids, which are DNA and also RNA. DNA is double-stranded polynucleotides, and they are found in the nucleus to store genetic information. Okay, and the sugar and the 5-carbon sugar that contains in DNA are the deoxyribose. Okay, the sugar found in the nucleotide of DNA are deoxyribose, and the bases are cytosine, guanine, thymine, and adenine. And then DNA is double-stranded. For RNA, the 5-carbon sugar is ribose, and the nitrogenous bases is cytosine, guanine, adenine, and uracil. There is no thymine in RNA. And RNA are usually single-stranded. There are three types of RNA that we are going to look at, which is mRNA, messenger RNA, tRNA, transfer RNA, and also rRNA, which is ribosomal RNA. All these RNAs involved in protein synthesis. The synthesis of protein is based from the instruction coded in the genes in the DNA. Synthesis and breakdown of polymers. Okay, so in order to synthesize polymers from monomers, the reaction or the reaction involves is called as dehydration reaction or condensation reaction. So this is where polymer are synthesized, a reaction that involves synthesizing polymers from monomers. Okay, so through the process of dehydration, in order to add new monomer to a polymer or to connect one monomer to another monomer is through the process of dehydration, where the reaction involves the removal of a water molecule. Okay, so the monomers will lose a hydroxyl group, and another monomer will lose a hydrogen atom. So this hydroxyl group and hydrogen atom will combine together to remove a water molecule to create or to synthesize a longer polymer. So for example here, the process of adding four monomers together to form a polymer consisting of four monomers involves the removal of basically one, two, three water molecules. Okay. And then once you have a polymer, the process of breaking down polymer into monomers, the process is hydrolysis. So the process of hydrolysis, from the name indicating hydro, it means that the reaction requires water molecule. Lysis, it means breaking chemical bonds, okay, between the monomers. So hydrolysis adds a water molecule, breaking a bond, breaking the bonds that connects monomers together. Okay, so in order to break the bonds between the monomers, a water molecule will be added, and hydrogen atom will be added to the monomer, and to one monomer, and a hydroxyl group will be added to another monomers, okay, as the results of breaking bonds between the monomers in a polymer. Okay, so that's it for this part. uh, See you in the next part of chapter 1.